Hello, my name is Chris Bunker. I'm the Senior Director of Business Development at Advanced Cell Diagnostics, which is part of Biotechne Corporation. Today, I'm going to give a presentation on our RNAscope in situ hybridization technology and its use in the detection of engineered cell therapies in solid tumor tissues. First, by way of introduction to Advanced Cell Diagnostics, we are again a part of Biotechne. We have about 200 employees in the San Francisco Bay Area. Our products and services are provided throughout US, Europe, and Asia. And our products are primarily probes. So we have nearly 25,000 probes in our catalog for the detection of essentially any RNA in fixed tissue, whether it's mRNA, long non-coding RNA, splice variants, even point mutations or microRNAs. We have probes and technologies for detecting any one of these. Our probes are provided either as products, kit-based products, or through use in our assay services. So just to provide some context for the assays that I'm going to speak about today, generally speaking, cell therapies are products based on T cells taken from a patient, engineered most oftentimes with a lentiviral vector, and then reintroduced into those patients to target antigens presented on certain tumor tissues. Historically, in the brief history of cell therapies, B cell therapies, B cell lymphomas and leukemias were the primary tumor type being targeted with cell therapies. And it's typically a fairly homogeneous population, primarily B cells in the tumor. And these can be analyzed somewhat effectively by flow cytometry, typically multi-parametric analysis to identify where are the T cells, engineered T cells, within that leukemic sample preparation. In contrast, solid tumors are much more complex tumor microenvironments consisting of many different cell types, and it's also very difficult to analyze those by flow. It's nearly impossible to detect a CAR T cell in that tumor tissue. It's certainly possible to look at immune cell populations with CD markers, but there are no effective means of looking at and finding where are CAR T cells or TCR-T cells in a solid tumor. To provide a little bit more context, the cell therapies, again, are typically based on lentiviral transduction. Lentiviruses are carrying either chimeric antigen receptors or T cell receptors that target antigens on a tumor population. While T cell therapies are the primary type of cell therapy, some other kinds that are being developed are macrophage-based or NK cell-based, mesenchymal stem cell-based, and gamma delta T cell-based. In all cases, they are going to be engineered to express either a CAR or a TCR. Now, again, the ability to detect those cells by immunochemistry or other protein-based detection methods is very difficult because that sample will, on the one hand, contain many antibodies, free-floating antibodies within the sample, which are very similar in sequence to the CAR being presented on a CAR T cell therapy. And likewise, there'll be many endogenous T cells that express T cell receptors that look very much like the engineered T cell receptors on TCR-T cells. So the solution that we provide is to detect those cells at the RNA level. Because the lentiviral based CARs and TCRs will almost always contain a UTR sequence that's derived from the virus itself, we now have a sequence tag that we can target with a probe to specifically detect engineered cells, whether they be based on any of the different cell types described here. As long as they can contain a lentiviral or synthetic sequence, we have a means of detecting those cells and differentiating them from any other cell in the sample type. That is the basis for all of the assays that I'll speak about in this. To summarize the features of the technology that I'm going to speak about through the rest of this presentation, the RNAscope in situ hybridization assay, it provides a means, again, of detecting mRNAs or any RNA sequence, including viral transgenes, in this case, as a means of detecting engineered cell therapies. The assay has sufficient sensitivity to detect individual molecules, meaning we can detect very rare events, very rare transcripts, and we can do that in a cell-specific manner. The assay can be multiplexed, 2, 3, 4-plex assays. It also can be combined with either immunohistochemistry or immunofluorescence, and it provides quantitative analysis. In addition, the RNAscope assay is clinically validated. It is being used for diagnostics in some cases, and it's being used very, very extensively in clinical trials as an exploratory biomarker, and also for pharmacodynamic and pharmacokinetic analysis of cell therapies. So the key features of the assay are the ability to detect RNA in formalin-fixed tissue, single molecule detection, and very, very high specificity. And this is based on the way in which the probes function to generate signal and amplify that signal in a tissue section. What's shown here and in the next few slides will be schematics of how we use oligonucleotide probes to generate target-specific signals. The probes themselves are simple oligonucleotides. The amplification module is a branched DNA-like structure consisting largely of oligonucleotides to which we then attach labels, either chromogenic enzymes or fluorophores for signal detection. These assays are all kit-based, meaning you can take a probe to any given target sequence, use that in conjunction with a kit, again, chromogenic or fluorescent-based detection kit. And these assays can be run on autostainers, Leica or Ventana autostainers, or they can also be run on a benchtop hybridization oven. The way in which we generate signal, again, is dependent on paired oligonucleotides binding to a target transcript and then hybridizing to other oligonucleotides for signal amplification. We have, again, over 25,000 probes in our catalog. We've validated all of these in silico. We have not validated all of them in wet lab testing, but we can be certain of their specificity simply by calculating the melting temperature of every oligo in every probe and being sure that it will hybridize specifically to the intended target and no other sequence in the species on which we're testing. Shown here is a schematic of a pair of the oligos that make up a probe. So each one of these oligos is hybridizing to about 25 nucleotides. The pair hybridizes to about 50 nucleotides. And the 5' ends, which we show as the top, will then hybridize to another oligonucleotide for amplification of signal. A given probe, whether it's to a CAR or TCR, interferon gamma, CD3, et cetera, or any other transcript, will typically consist of a pool of oligonucleotides spanning about 1 kb of sequence. And that means it'll consist of about 20 of these pairs. Each one of these pairs can generate signal, but we only need very few of these pairs to actually generate detectable signal. So by using 20 or so oligo pairs in a probe, we have a very highly redundant method of generating signal for single molecule detection. And that's a feature which is particularly applicable to the detection of RNA in formalin-fixed tissue, where the target sequence will be highly fragmented. It'll be bound by proteins. There'll be many elements which will, or many factors, I should say, which will impair the ability of these oligos to bind. Nonetheless, as long as a couple of them can bind, there will be a sufficient signal generated to detect that molecule as a dot, a visible dot in the tissue. Now, the RNAscope assay is our primary assay for mRNA detection. It has a variety of features, most notably the fact that we can multiplex this, 2-plex assays for chromogenic detection, 3 or 4-plex assays with fluorescent detection. We have some higher plex assays that are suitable for frozen tissues. And then the assay is being used for diagnostic applications as well as as a research tool. I should say there are no FDA-approved diagnostics yet, but it is actually being used for diagnosis, particularly for HPV and head and neck cancers. The other assay we have is called BaseScope, and this is also based on the same principle of paired oligonucleotides binding to a sequence to generate an amplified signal. But in this case, we use slightly different probe chemistry, probes that are more like locked nucleic acids and therefore have much higher affinity. And with these, we can design a probe which will even differentiate a point mutation from a wild type sequence. And in addition, we use a much larger amplification module such that a single pair of oligos will generate detectable signal. As compared to RNAscope, where we need a few of all of those pairs to bind to generate detectable signal, a single pair of oligos with BaseScope will generate a visible signal. And so now we can target either very short sequences like pre-microRNAs, single guide RNAs. We can target exon-exon junctions. We can detect point mutations, TCR clones, etc. So these are the two assays we have available for mRNA detection and variant mRNA sequences using base. Here I'll show schematically how signal is amplified with this paired oligonucleotide approach. With oligos hybridizing to a target transcript, they will then be able to stably hybridize to this preamplification oligonucleotide, which then hybridizes to amplifier oligonucleotides, which then hybridize to either chromogenically labeled or fluorescently labeled oligonucleotides to generate signal. And this assay, again, allows for localized amplification that is on the order of 1,000-fold per pair of oligos, or over 5,000-fold for paired BaseScope oligonucleotides. And this happens in a way which is, again, dependent on multiple oligonucleotides hybridizing simultaneously to a target sequence, which is how we're able to generate such high levels of signal without any background signal coming from non-specific binding. So to show you an example and illustrate how we're able to detect single molecules of RNA in fixed tissue, the assay shown here is performed on HeLa cells, where RNAscope was used to detect HER2 transcript in the left-hand experiment, an image, and on the right-hand side, HER2 transcript was measured in solution with a quantitative solution-based assay where a standard curve of in vitro transcribe message could be included. And the outcome of this comparison is showing that when we count, in this case, green dots corresponding to HER2 transcript from an RNAscope assay, we get about 14 dots per cell in a field of view. And in comparison, if we quantify HER2 transcript in solution, we get about 17 copies on average from extracted RNAs. So the primary conclusion here is that a dot corresponds to a single molecule of RNA. Likewise, if we take these same probes used here on HeLa cells, in this case to detect RNA, now we use more extensive permeabilization conditions to allow for exposure of HER2 DNA. The same probe generates 2 dots in a HeLa cell, which correspond to the 2 wild-type loci, the 2 genomic loci in the HeLa cells. In comparison, we now see multiple dots if we use that same probe on SKBR3s, which are amplified for the HER2 locus. So this again further emphasizes or illustrates the fact that the signals we're detecting are individual molecules, in this case, 2 different genomic loci, in the previous example, multiple HER2 transcripts. Now we like to emphasize the fact that our assay provides more information than extraction-based methodologies. And here I'm going to try to illustrate that by comparing RNAscope with a very well-established, very sensitive methodology, NanoString, which is also able to detect and measure transcripts in formalin-fixed tissue. However, NanoString just gives you a single value per tissue sample for a given transcript, whereas the RNAscope assay will give you information about both expression level as well as number of cells that are expressing a given transcript. So in this case, if we look at, for instance, TNF-alpha, IL-1 beta, or CTLA-4, the average expression of these 3 markers in these non-small cell lung cancer tissues is less than 1 copy per cell, given the fact that there are hundreds of thousands of cells in the tumor and only a very few cells expressing. The average value ends up being very, very low, whereas if we measure this with a spatial view of expression using RNAscope, we see that there are very few cells. We can quantify, count these cells, and we can measure the expression level of each one of these transcripts in that cell population. So here's an example of low copy transcripts for endogenous messages. We can just as well apply the methodology to count CAR T cells, for instance, or any other sort of cell therapeutic, measure the number of cells, and then also measure the expression level within those cells. And we typically use Halo software. There are other softwares that are available to do the same type of analysis, but we typically use Halo from Indica Labs as a means of identifying cells that are positive for expression for any given transcript. And with Halo software, we can quantify expression of single markers, 2, 3, or even 4 markers to look at expression level and percentage of cells that are positive for 1, 2, and 3 markers. Now, the ability of RNAscope to detect individual transcripts and make measurements of transcripts within individual cells in the spatial context of intact tissue morphology is what enables us to provide a very useful means of detecting and measuring cell therapies in patient tumor biopsies. I spoke previously about the fact that the CAR or TCR transcript will typically have viral or synthetic UTR sequences, and that we can use these sequences as a means of designing specific probes that will identify those cells and differentiate them from any other endogenous cell. And so that's the approach we've taken in many different cases. And again, in comparison to protein-based methodologies like IHC, our probes work effectively for CAR T-cell detection because of the unique sequences that are present in the transcript. IHC is not really effective because there are no unique epitopes or antigens on a CAR protein to differentiate it from other endogenous proteins or cells in the sample. In the next few slides, I'll show examples of use of RNAscope for measurement of cell therapies being used in a number of different clinical trials for solid tumor treatments. This is work from Novartis and UPenn, which was in relation to their EGFRB3 CAR T-cell program in glioblastoma. And what's shown here are biopsies and assays run on biopsies taken from either pre-treatment or post-treatment with their CAR T-cells. And what's shown in the middle panel is the RNAscope assay to measure the CAR T-cell presence in the tumor following treatment. On the right-hand side is another RNAscope assay used to measure the activation. In this case, it's measuring interferon gamma as an activation marker to show engagement of the EGFRB3 target. And then in the left-hand panel, CD3 IHC to show recruitment of T cells following CAR T-cell target engagement. This is an example of RNAscope applied to detect TCR-T cells. In this case, a liposarcoma patient biopsy taken post-treatment with a TCR-T cell targeting NY-ESO. Here again, we use a probe targeting a UTR sequence of that TCR-T cell. And this was combined with an RNAscope probe for detection of CD3 to simultaneously measure the cell therapy as well as recruitment of endogenous T cells into the tumor. Here again, same story, different scenario. In this case, CAR T cells targeting PSCA expressed in pancreatic cancer, and we've combined the detection of the CAR T cells by RNAscope with detection of CD3 positive T cells by IHC. RNAscope can also be used in a preclinical setting to both evaluate CAR T cell activity and efficacy in animal models, but also being used to look at the safety of CAR T cell targets. And that's illustrated in work that we did in collaboration with Bluebird Bio, in this case in relation to a target RR1. Now, the backstory is that RR1 was known to be expressed in human lung at very, very low levels, and the concern was whether this expression level might lead to adverse events if a CAR T cell targeting RR1 was taken into the clinic. So the preclinical study was done in mouse lung, where the question was, is there RR1 expression in the mouse lung, and will that result in any sort of adverse events if those mice are treated with CAR T cells targeting RR1? So the observation was that there is very, very low level expression in mouse lung, specific cells, endothelial cells of the pulmonary vein. And this low level expression can be seen here as a few dots with the RNAscope assay, but this expression was undetectable by IHC. However, the phenotype that results from these mice being treated with CAR T cells, which target RO1, was pulmonary toxicity. And so what's shown here is CAR T cells infiltrating into the lung based on an RNAscope assay detecting them, and then the expression of granzyme B, a marker of T cell activation, which relates to the resulting pulmonary toxicity. So despite the fact that the target was undetectable by IHC, the RNAscope assay detected signal and expression, which correlates with CAR T cell recruitment and pulmonary toxicity. Another interesting observation made in this mouse model was that xenograft tumors that also expressed RO1 did not show any response to CAR T cell treatment. That's shown in this graph here, where the yellow curve corresponds to the tumor volume of RO1 positive xenograft treated with CAR T cells and showing no response, namely tumor volume continues to increase just like vehicle-treated animals. And that differs dramatically from another mouse model of BCMA, where the xenograft tumor which expressed BCMA was responsive to anti-BCMA CAR T cell treatment in that the tumor volume went to zero, effectively killing the tumor. So the fact that there was response in the lung to anti-RO1 CAR T cell treatment, namely pulmonary toxicity, but no response in the xenograft tumor, indicates that perhaps the CAR T cells were either exhausted upon activation in the lung or never were recruited to the xenograft tumor following their activation in the lung. This can also be seen at the molecular level, showing the fact that the xenograft tumor expressing RO1, expression of RO1 being shown on the right, show no infiltration of CAR T cells, as shown in the panel on the left, where there's no red signal corresponding to the CAR T UTR probe. There is some low-level expression of granzyme B, but nothing to do with CAR T cell activity. And that's in contrast to the situation with the BCMA positive xenograft tumor in a different mouse, where here we're showing BCMA expression as measured by RNAscope on the right, and we're seeing CAR T cell infiltration into that tumor as measured by the signal in red on the left, and also activation of those CAR T cells as illustrated by interferon gamma expression from those and other adjacent cells. Just to illustrate what we can do with more highly multiplexed RNAscope assays, in that same model, we ran a 4-plex configuration where we looked simultaneously at CAR T cell detection with an RNAscope probe, as well as activation markers, granzyme B and interferon gamma, also measured by RNAscope, as well as the population of CD3 positive T cells as measured by an immunofluorescent assay. So this level of plexing is possible with either RNAscope alone in a 4-plex configuration or combinations of ISH and IHC or ISH and immunofluorescence. I would just like to reemphasize the fact that all the assays that I've shown in the previous slides and assays with the other 25,000 probes in our catalog, as well as any other assays we design going forward, are all designed based on the same principle. And as a result, the oligonucleotides in those probes all have essentially identical affinities, all of which enable single molecule detection and all have the same very high specificity. This is a feature of the assay that we're able to design probes for detection of virtually any sequence, and they can all be put to use immediately upon design. So in addition to being used in the ways I've shown in the previous slides for cell therapy detection, they are also used very extensively for use of in biomarker assays and for target detection, targets of both CAR T cell therapies as well as many other types of therapeutics. Just to give you a few examples of CAR T cell and TCR-T cell therapeutic target assays, here are some targets for which many cell therapy programs are being developed and for which the targets are very difficult to detect by more conventional methods like IHC. NY-ESO, mesothelin, HPV16, E67 transcripts, and PSCA are all targets for which IHC is not very effective in terms of specificity or sensitivity. All of these targets are detected very effectively with the RNAscope assay. And just to where RNAscope has been proven to be effective, in fact, more effective than any other methodology. I'll show you a couple examples where it's been used as a biomarker assay, not in cell therapy context, but in other therapeutic developments. Here, work that we did with Insight on their IDO1 program. And here, what we showed in a very short period of time was that RNAscope was substantially more sensitive, in fact, 10 times more sensitive than the IHC assay, which they'd invested heavily in and spent a great deal of time trying to develop before discovering RNAscope and finding that we could provide them with an assay which was, again, about 10 times more sensitive than the IHC assay. Another example was a biomarker assay we developed for Merrimack for their anti-ERB3 therapeutic program. In this case, the biomarker that they identified was most effective in identifying patients was the ligand of ERB3, which is heregulin. This is a secreted protein, so IHC was essentially out of the question. They were not going to be able to reliably detect heregulin expression by IHC in these tumor biopsies. And it turned out, based on comparison, that RNAscope showed superiority in terms of both sensitivity and specificity compared to qPCR. They were not going to get enough material from a patient in a needle biopsy to reliably detect heregulin message by qPCR, whereas a very small amount of material was sufficient to allow for cell-specific detection in tumors within that needle biopsy by the RNAscope method. Another example is an assay that we developed with Bayer Pharmaceuticals for their pan FGFR123 inhibitor, where, again, in comparison to other more well-established methods, IHC and nanostring, we were able to show them that an RNAscope assay was both more sensitive and more specific than these other methodologies. So, and again, as in the case of Insight and with Merrimack, we can design probes very quickly. We can show their performance very quickly and provide a means of comparing assay performance to other methodologies. And here, again, the outcome was higher specificity and sensitivity to other methodologies, which led them to choose the RNAscope assay for companion diagnostic development with our partner Leica Biosystems. So these are just illustrations of how RNAscope compares to other methodologies with an emphasis on the fact that we can design probes and demonstrate their performance very, very quickly as compared to the amount of time it can take for other types of assay development. So just to review, we have two assay technologies, RNAscope and BaseScope, both of which are RNA in situ hybridization methodologies. And with these, we can detect essentially any RNA in fixed tissue. We can either detect mRNAs, we can detect splice variants, we can detect point mutations. Any of these can be detected with equally high specificity and sensitivity with one or the other one of these assays. And while we are primarily a products business providing probes and detection reagents to our customers for use in their labs, we also are a service provider. Our Pharma Assay Services Lab supports a great deal of research, particularly in cell therapy, particularly supporting clinical trials. We have a great deal of capacity with a large number of autostainers, which run pretty much 24/7. We have the ability to do analysis with both Halo software as well as a board-certified pathologist. And we also have a tissue bank for doing retrospective studies on to look at target expression and for biomarker development. What I presented today is the RNAscope technology as a tool, essentially the only tool by which to detect and characterize cell therapies in solid tumor tissues. We can perform tissue-based analysis of any sort of cell therapy based on viral vector detection, as well as detection of any other mRNAs that are relevant to understanding cell therapy activity in a tissue biopsy. The assays we provide in all cases enable single molecule detection with single cell specificity in the context of a spatial analysis of a tumor microenvironment. These assays can be multiplexed and they can be quantified, quantifying 1, 2, 3, or 4 markers simultaneously. We are supporting many cell therapy programs that are going into phase 1 clinical trials with assays that will characterize the performance and activity of those cell therapies in post-treatment patient biopsies. I hope this information has been useful to you, and I thank you very much for your attention.